organizers for inviting me here today and giving me the opportunity to present some of my work. So, as you all know, in a typical eukaryotic cell, about 99% of the DNA is wrapped by nucleosomes. The remaining 1% of the DNA is distributed into about 100,000 distinct loci, and these are known as open chromatin regions, or DNA one hypersensitive sites, or DHSs for short. The pattern of DHSs is cell type specific, and it's an important determinant of cellular identity. The reason for this is because nucleosomes will prevent DNA transcription factors and other DNA binding molecules, and thus they form an important part of the regulatory mechanisms. So what we'd like to do ideally in the long run is to understand how and why the DHSs patterns that we observe in each cell type are established. However, I will only make a very short and a very small attempt towards that big question by categorizing the patterns of DHS specificity that we observe and try to present a simple phenomenological generative model that could explain what we're seeing. So to set up the scenario, here's the cartoon. We see 3 different cell types, and we see the DHSs that are marked as colored circles. I'm interested in the overlap between different cell types, and I will define overlap between 2 DHSs if they share at least one base pairs in these different cell types. And I will be characterizing each DHS by the number of cell types where it's present, which I'll be referring to as the specificity. As you can see here, you have specificity 3, 1, or 2 for each of these different DHSs. I will also be referring to the ones that, the DHSs that are found in many different cell types as ubiquitous, and the ones that are found in only one cell type as specific. So thanks to the ENCODE project, we now have lots of data from many, many different human cell types, which allows us to analyze this kind of questions in a large-scale fashion. So what I did was to download DHS data from 72 different cell types, and I was able to identify 1 million, roughly 1 million unique non-overlapping loci, and all of them were found at least 5 kb away from any annotated TSS. So here I'm showing you an example of this type of data, and what you can see over here, I cannot see at all. Oh, here it is. Here is a ubiquitous DHS. It's found in all 72 cell types. Over here we have a specific one. It's only found in one cell type. And over here we have 2 intermediate examples. If we crunch the numbers, for all of the 1 million DHSs, we found a distribution, which is shown here by this blue line. And as you can see, we have only more than half of the DHSs are found in only 1 or 2 cell types. This is not at all consistent with what you expect from a binomial model shown here in green. The binomial model is what you'd expect. A binomial distribution is what you'd expect if the probability of overlap between any 2 cell types was a constant. Instead, we find a much better fit to the black line, which represents a power law distribution with a slope of minus 1.35. So in addition to looking at lots of human cell types, ENCODE has also looked at DHSs in mouse cell types. And we find, as was mentioned yesterday, that DHS specificity appears to be a conserved trait. This you can see here in this heat map, where we have mouse on the y-axis and human specificity on the x-axis. And as you can see here in the dark red, DHSs that are highly specific in mouse also are typically highly specific in human. And over here, we see that we have an enrichment for things that are ubiquitous in both mouse and human. We also found that the underlying sequences at these DHSs were more conserved in the ubiquitous DHSs compared to the specific one. So this suggests that there is some sort of selective advantage that for these ubiquitous DHSs, which allows them to be more conserved than you would expect from the binomial model. So to understand how this works, I'm going to propose a very simple model. And here I'm going to be illustrating this in the next part of this talk. So here we consider one cell type, which has 5 DHSs labeled A, B, C, D, and E. We assume that over evolutionary time, this cell type attains the ability to differentiate into 2 different other cell types. These 2 other cell types have their own set of DHSs. And the DHSs that are found in the children is determined by the parent. So the rule that I'm using is that each DHS in the parent is copied into the children with a probability which is given by this equation here. Here, K represents the specificity, the number of cell types where the DHS is present, and B is a parameter, which I'll be referring to as the bias or the selective advantage. So in this example here, you can see that A and B both got copied into the children. C and D only made it into one child each. And what I also did here, for each child, I added 2 new DHSs so that they still have a total of 5. I continue running. In the next step, I do the same procedure by choosing one of the children at random, and then I just run the same procedure. I also update this master table here, which keeps track of the specificity of each DHS. I keep running this procedure until the tree has reached a pre-specified size of C nodes, where C corresponds to the total number of cell types in the organisms. At the end of this procedure, I will sample from the tree 72 nodes, which will allow me to compare directly to the data. So when I run this scheme, you can see the data in red compared to the model in blue. The parameters that I used was to assume that the number of DHSs in each cell type is drawn from a normal distribution with a mean of 100,000 and a standard deviation of 20,000. I assumed that there were a total of 1,000 cell types. I sampled 72 of them, and I repeated the whole thing 10 times to give me error bars. As you can see, there's a rather good correspondence between the model and the data. Finally, we have this parameter B. If you do some analytical analysis of this random binary branching process, you can show, after several simplifications, that the slope of this power law should correspond to the selective bias B. So I used the value 1.35, which I had estimated from the data, and when I measure the slope from my model, I get a value of 1.4. This clearly suggests that, at least to a first-order approximation, this very simple model seems to be capturing what's going on in the data. So next, I try to use this model to address other related questions to DHS overlap. So going back to this cartoon here, I can define pairwise overlap between any pairs of cell type based on the number of DHSs that they share. So in this cartoon, you can see that the blue and the green share 3 out of 4 DHSs, so this means that the overlap is 75%. I can calculate this overlap between all the pairs of 72 cell types that I found in the data, and then I can use this overlap to cluster them. And this is shown here in this dendrogram. So I don't expect all the people, especially in the back, to be able to read all the labels down here, but I'll tell you some of the salient features about this dendrogram. So over here to the left, we have a tight cluster, which seems to separate pretty quickly from everything else. It turns out that all of these correspond to blood cells. And here we also find the highest degree of overlap, which is 86%, which is between a neonatal and an adult blood cell type. Another way of representing this data is by using a distance matrix like this. And here I've colored, the colors correspond to the relative overlap. And as you can see, we span all the way from almost 90% down to 10%. And here, the orders on the x-axis, on the y-axis, are the same as in the dendrogram on the page before. So the fact that we observe these big swaths of blue and other fancy colors is not surprising. So to orient you here, again, here is the tight cluster of blood cells that I found before that all appear to be very similar. Over here, we have a cluster of cells which appear to have a lot of blue, which means that they sort of don't look like anything else. And by inspecting the cell types, it suggests to me that this corresponds to a bunch of different cell lines, because it includes K562, JERCAT, and MCF7. Finally, this blob here in the middle appears to correspond to various tissue samples from the human body. So what I can do is that I can take all of these overlaps that I measured in the matrix on the slide before, and I can calculate a cumulative distribution function. And I can do the same thing for the model that I had. And as you can see, again, the correspondence between the model and the data is very nice. The main discrepancy is here on the left-hand tail. And the fact that the blue is above the green suggests that we have a lot more of cell types with very low overlap in the model compared to the data. So we can use this type of DHS overlap to try to quantify the notion of cell type similarity, and assign a distance number to the similarity. So the way I define cell type distance is based on this tree here, And here I filled in numbers on each node, and they represent the distance, which is just the number of steps from this cell type, which is marked in red here. So for the model, this is very easy to calculate because I have full knowledge of the topology of this tree. And so I plotted here the distribution of the average overlap as a function of distance. And as you can see, it's roughly a linear function, and it decays rather slowly. So this suggests that if you have an overlap between two pairs of cells, which is 50%, it suggests that they're typically almost 10 nodes away from one another in this tree. Finally, we have also analyzed the DHS specificity and tried to correlate it to numerous other genomic properties, and we find that the specificity does indeed correlate with several of them. Here I'm showing you one example. I calculated the distance to the nearest TSS for each DHS, and then I average based on the specificity. And as you can see here, the ubiquitous DHSs are on average much, much closer than the specific ones. So finally, I would just like to say that I wrote a rather ambitious abstract, and I was hoping to cover a lot of stuff about transcription factors, which show some interesting patterns related to the specificity. But I just did not have time to squeeze that into this short talk here. So I hope that instead of calling your lawyers and filing a false advertisement lawsuit against me, that you can come up and talk to me about it afterwards. So to summarize, I've characterized the specificity of DHSs across multiple cell types and found that it constitutes a power law. I presented a simple phenomenological generative model, which is based on only one simple principle, that the parent is used as a template. Only one equation, which is shown here. And this equation is motivated by the fact that the DHSs that are found in multiple cell types are probably more likely to be very useful, and hence, they're more likely to be reused in the daughters. And finally, the model only has one parameter, which can easily be related to experimental variables. And as I mentioned very briefly, we can relate the specificity to various genomic properties, which in turn can be related to the function of, or presumed function of these DHSs. So on the last slide, I'd like to thank my supervisor, Dr. Gabriel Krayman, and several other members of the lab who helped me out with this type of work. I'd like to thank you for your attention, and I'll take any questions.
That was a really interesting approach. I'm wondering how you classified overlap, given that you start off with different sizes of DNA hypersensitive sites.
Sorry, I classify overlap?
Given that the DNA hypersensitive sites are different sizes, and you could have two overlapping one, or...
Sorry, how I...
Like, let's say you had one big hypersensitive site,
yeah,
and in a different cell line it's only overlapped partially. Would that be a full overlap? And
yeah, so let me show you the... So basically, you don't require all of the DHSs to overlap. So here you see these two guys don't overlap, right? But you basically, these two guys here, but you basically find the convex hull of a set of sites that overlap, and you take the maximum of that.
So I'm wondering why all your sites are the same size.
That's because they were like that in the ENCODE data that I downloaded. I think I looked at it, and maybe someone from ENCODE can tell me why that is, but very few of them differ from the median.
Next question.
Thanks for your talk. So I noticed that the branching process you use to model the cell types is very similar to the coalescing for demography. I'm wondering whether it's possible to borrow some existing theories worked out for the coalescent models.
If you know anything about those things, please, I'll be very happy to hear about them. That's a great suggestion.
I have a question. So does that hierarchical clustering show any kind of pattern of development? So if you have I know that you have some embryonic cells in ENCODE, so and the ones and some further down in development. So is there DNA hypersensitive sites that disappear or appear as this progression?
I haven't analyzed it from that point of view. I asked Lampanacchio yesterday because I don't know the relative order, temporal order between these two cell types. And he said that when he looked at the 7 cell types, that he would rarely see something which appeared to come up early and then come up at, say, the 7th time point. So I don't know if that's the case or not.
Okay. Thank you.
So maybe I'll inject one last question, if that's okay. So, you know, I mean, one way of asking my question is, what does it all mean? And I can try to be more specific, right? I mean, there is a process of evolution underlying the whole thing, right? So you'd have, some of this would be neutral, some of this would be functional. Can you start seeing the different categories, maybe, of switches between when DHSs come and go, or between cell types that you have there? Does that make sense?
I mean, that's what I would like to be able to see. I haven't been able to spot any like that. But
right. Yeah. Okay, so our next